We now come to the second talk, and it gives me great pleasure to invite our next speaker, Dr. Yoko Fukushima, who has come all the way from Osaka University in Japan. A celebrated researcher, she has several publications in hardcore research. She has won several awards, including Medical Research Award from Japan Medical Association, the Roto Award, Bayer Retina Award, the Joss Young Investigator Award, and Japanese Association of Pediatric Ophthalmology Award. Welcome, Dr. Yoko, and I request her to talk on intrinsic and extrinsic factors regulating retinal vascular pathfinding during development. Dr. Yoko.
Hello everyone, my name is Yoko Fukushima from the Department of Ophthalmology, Osaka University, Japan. It's a great honor to be here. I appreciate the opportunity to give this presentation. Today, I would like to talk about the three topics. In the first two sections, I'm going to share the findings obtained from our previous research. Based on that, I would like to discuss with you how we should use anti-VEGF drug for ROP. I hope these topics will provide a better understanding of the pathophysiology. Let me start with the first question. What regulates physiological retinal angiogenesis? These images show vascular development in postnatal mouse retinas. You can see highly organized blood vessels are built over time under physiological conditions. Intraretinal angiogenesis is supposed to require three factors. The first is extracellular matrix. This image shows the astrocyte network in developing mouse retina. The pattern of growing blood vessel in red corresponds with astrocyte network in green. The extracellular matrix is secreted by the astrocyte and serves as a scaffold for the vascular network. The second is extracellular signaling molecules of blood vessel, which are mainly composed of secretory proteins. Proangiogenic molecules such as VEGF promote expansion, branching, and dilation of blood vessels. On the other hand, anti-angiogenic molecules such as semaphorin 3E, called SEMA3E, inhibit those vascular changes. The third is intracellular signaling molecule. Let's have a look at the video. Endothelial cells sense the surrounding environment and migrate to oxygen-demanding area through extracellular dynamic change of cytoskeleton. This directional movement of the cells is regulated by extra signaling molecules. Here you can see on the right one of the intracellular molecule, ROJ, a member of small GTPases. This live imaging is from the aorta ring assay. We labeled endothelial cells with different colors. In wild-type cells on the left, they are moving forwards. However, ROJ deficient cells on the right behave in a confused manner. So during angiogenesis, intracellular molecules make a decision about which direction to move. The answer to question 1, physiological angiogenesis, is regulated by scaffold, extrinsic factors, and intrinsic factors. In contrast, ROP, the blood vessel, grow into the vitreous and not into the retina. The three factors could change in terms of quality and quantity in ROP. Can we control the direction of vascular growth rather than just controlling abnormal blood vessels? The oxygen-induced retinopathy model, which called OIR model, has been used for analysis of pathological angiogenesis. In this model, after the regression of capillaries, the arising hypoxia induces vascular regrowth to ischemic retinas. Abnormal vessels are eventually formed as seen in human ROP. The VEGF level in pathological conditions was significantly higher than those in physiological conditions. That's why anti-VEGF drugs are effective for the inhibition of abnormal blood vessels. However, do we have any options to control these blood vessels by manipulating signaling molecules? I'll give you an example of intracellular molecules, ROJ, as I mentioned earlier. In the OIR model in which VEGF overexpressed, ROJ deficiency inhibited forward movement of endothelial cells and dramatically suppressed abnormal blood vessels. How about extrinsic signaling molecules? When we injected the extracellular molecule SEMA3E protein in the vitreous of OIR model, it succeeded in preventing abnormal vessels and promoting vascular regrowth. This is promising for treatment. But please take a look at the image on the right. The anti-VEGF drug reduced pre-existing intracellular capillaries. This data seems to suggest that we will be able to manage the direction of human retinal vascular growth in the future. I believe that anti-VEGF drugs have dramatically changed ROP management. The number of successfully treated infants is likely to continue increasing. However, we are still concerned about the potential risk of the drugs. The retinal image on the left shows trotus and dilated vessels and vascular loops and a hemorrhage in the vascularized retina. The baby was diagnosed with aggressive ROP. I treated her with ranibizumab. As you can see on the right, 4 weeks after treatment, retinal blood vessels were thinner and vascular growth had stopped. It reminds me of the mouse retina treated with VEGF trap. I'm thinking if the timing was too early to treat, maybe not, but it's still on my mind. Also, these images may give you concerns about anti-VEGF therapy. As shown in the image on the left, I've seen this kind of atrophic retinas after anti-VEGF therapy in a few infants. In addition, I have observed vascular growth arrest and vessel thinning. I'm worried about the potential adverse events due to suppression of VEGF in developing retinas. Anti-VEGF therapy has absolutely improved the management of ROP, but we should be aware of a few concerns about neurons and vascular abnormalities. We need to consider what is the appropriate dose, timing, and agents of anti-VEGF drugs. In addition to anti-VEGF drugs, I hope drug discovery targeting other signaling molecules in the future will provide more choices to normalize retinal angiogenesis in ROP infants. Thank you very much.
Excellent talk, Dr. Yoko, and thank you for finishing ahead of time. I'm sure it's going to generate a lot of discussion. It's really interesting to see that you can actually manipulate the direction of the blood vessel growth. Thank you so much.